So hi everyone, my name is Haoyi Qiu. I am a PhD student in the Department of Physiology at McGill University under the supervision of Dr. Reza Sharifnaini. So today I'll be talking a bit about my research, the spinal mechanisms of pain. So my research is very specific on one subset of neurons in pain, but today I'll talk a bit more about the importance of spinal cord circuitry and how these neurons talk to each other and how this leads to our perception of touch, pain, different sensations. And if something goes wrong with this spinal circuitry and how these neurons communicate, then that's how we can get disorders like chronic pain. So I'll start with a bit about me. So I grew up in Calgary, Alberta, and then I came to McGill University in 2017 to start my bachelor's, and my bachelor's was in physiology and mathematics. So the reason I chose physiology and mathematics was because I really enjoyed math in school. I enjoyed like the logic behind the math and how in most math questions, you really just have one correct answer. But physiology really fascinated me. I found the human body really, really interesting, but my physiology courses were much more difficult for me than my mathematical courses. And I did do a bit of mathematical research, like I did some computational work where I joined a lab and we did some modeling of like biology, biological diseases and everything. And I also joined the lab of Reza Sharifnaini in my undergrad, where their lab mainly studies chronic pain. And this was really interesting to me because I know that pain is something that is very like nobody wants to be in pain, and you take these pain medications to alleviate your pain. But we were looking at sort of a different approach to pain where you're understanding why does the body feel pain. And to me, this was really fascinating because I have never thought of pain in that aspect of like, okay, like what is actually changing in this body that's making this person be in pain. And we have the International Association for the Study of Pain that has a textbook definition of what is pain, which is an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in terms of such damage. So this is what they define pain as. However, you can see that pain is a very internalized experience, and it can affect people of all different ages. So you have children that are living with pain or chronic pain, and you have elderly and people of different colors and races. So it doesn't affect only one type of race or ethnicity of a person, and it affects everyone, and also different genders as well. So you can see here that there's a low back pain. There's also like headache pain, facial pain, stomach pain, so gastrointestinal pain, toothaches, and you can also have like knee pain or like osteoarthritis. So pain is acute pain helps us survive, but chronic pain is when it becomes a disease. So this is pain that carries on for longer than 12 weeks despite medical or medication or treatment. And there's many different types of chronic pain. So it can be in the form of inflammatory chronic pain, musculoskeletal chronic pain, and also neuropathic pain. So there's this website called the Invisible Project that sort of highlights patient stories dealing with chronic pain. And I invite you to read some of their stories. So this is a 16-year-old Amira from Arizona, and she lives with a type of neuropathic pain called trigeminal neuralgia, and she has chronic oral facial pain. And she is 16-year-old, so she's still in high school. She's trying to navigate the different, being in high school and also dealing with chronic pain. And one of her symptoms is cold allodynia, where sort of wind blowing on her face makes it really painful, and it's a really debilitating type of pain for her. So they highlight many different patient stories, and you can see that pain affects everyone, and it's not just a specific type of person, and it's very universal in that it doesn't discriminate, and anyone could be in dealing with chronic pain. Because for me as a researcher, I work in the lab with animal models. I don't interact with patients a lot. I like to use these resources, and I also like to interact with to talk to patients at different conferences and events like this to hear more about their battle with chronic pain, to sort of put a more humane perspective on why I'm doing this research. So what I study is something called nociception. It's the neural encoding of noxious or pain-like stimuli. So because I work with animal models, it's hard to actually ask the animal to rate its pain on a pain scale. It's not like human patients where they can fill out questionnaires and that can give me an idea of what kind of pain that they're feeling. It's really hard to do that with animal models. So I look at something called nociception, where I look at the neural activity of these animals, and I'm able to tell, to see what the neuron responds to a pain-like stimuli, and that can give me an idea of how this neuron is communicating to its neighboring neurons and transmitting this pain information. So the pain processing pathway is shown here. It starts, this is how the central nervous system, so the spinal cord and the brain, is able to perceive a painful stimuli like a needle pricking your finger, how it travels through the spinal cord and towards the brain, so that you're aware of this painful stimuli. So you can see here that the first step is the transduction of pain. So this is how this mechanical sort of indentation of a needle on your finger gets translated to a electrical signal from your neurons that then gets transmitted along this first order neuron towards the dorsal root ganglia and into the spinal cord dorsal horn. And it's here where it synapses to a second order neuron that then is able to bring this information towards the ascending up the spinal cord and into the brain, where you can then have a descending modulation. So the brain can then send information back to the spinal cord to sort of silence the pain information. And also it can also send information down towards like motor neurons for you to then remove your finger away from the pain. So there's many different steps in the, or sort of levels of perception in the pain processing pathway, from how does a neuron from your finger that actually perceives is getting the pain information, to how it travels all the way to the brain and how your brain realizes you're in pain. So I'm especially interested in the spinal cord area and how sort of these peripheral neurons carrying information from your environment, what happens when it brings this information into the spinal cord, and how does the spinal cord choose which type of information to send to the brain? Because, as you're sitting here, you're receiving many different stimuli from your environment that you might not be noticing, like perhaps you're sitting in a chair, perhaps you're outside where there's wind. There's a lot of different stimuli happening, and how does your spinal cord sort of filter out what's happening and only choose to send certain information to your brain? So one of the theories proposed Patrick Wall and Ronald Melzack in the 1960s is this gate control theory. So in the substantia gelatinosa of the dorsal horn, so this is an area of the dorsal horn, there contains sort of a gate, a neuron gate, that can either be opened or closed, and this can block or transmit pain information. So you can see here, in the normal condition, when there is a closed gate, there's more activity from these large diameter fibers. So So you have these large diameter sensory fibers that carry mainly touch information, and they can activate these, the sort of gatekeeper, which is an inhibitory neuron in the dorsal horn, and they prevent pain information from being transmitted to the brain. Now, if this gate is open, now you have pain information being carried by the small diameter sensory neuron that is just free flowing and activating these pain transmission neurons and sending pain information to the brain. So this gate is sort of silenced, and this gatekeeper is sort of silenced. So now this gate is open, and you have this touch information, or this pain information sending all the way to the brain. So this is sort of the basis of my project. So this idea was proposed in the 1960s. And since then, we have refined this theory a lot more. And we are looking at specific populations of neurons and trying to define, like, how do we selectively target these gatekeeping neurons, and what other molecular identities do they have? So a bit about like what I use to study pain. So I mainly use animal models. So these are in the form of mice. We have a couple transgenic mouse lines. So this is where we are able to use these mice to only look at a certain population of neurons. And for me, I have a transgenic mouse line that is able to help me visually target only a small population of inhibitory neurons in the dorsal horn. So this is very useful when I want to do recordings on these specific neurons or look at their molecular identities. I can actually visually see these neurons, so it's very I can then do experiments with them. So other techniques we use in the lab are cell culture. So we're able to extract tissue from these animals and then culture the neurons in a dish, and then do experiments like recording from these neurons or examining the molecular identities of these neurons. And it's very accessible because these neurons are in a dish instead of in an organism. So something else that I do, so this here is an extracted spinal cord, and I'm able to dissect out the spinal cord, and it's still living. So I keep it in a fluid that's able to help these neurons survive. And I'm able to do recordings directly in the neurons from the spinal cord sections. And this way, the circuitry of the spinal cord is still intact, because when we do cell culture, we sort of dissociate the neurons so that it's just an individual neurons in the dish, and they're not connected to the original circuitry that's found in the organism. But when I extract out the spinal cord like this, and I just take slices, the circuitry of the spinal cord is still intact. So I'm able to see which neurons connect to which neurons and how they're talking to their neighboring neurons. So this is all currently being done in mice, but our lab is very fortunate that we get to work with a team of neurosurgeons that are able to extract human spinal cord tissue from donors. So when there is a transplant donor, many of the organs get taken for transplant, and we are also able to take the spinal cord and bring it back to the lab and do experiments on it. So this here is spinal cord tissue that has been preserved in ice, and it's been frozen. So I can take sections of this human spinal cord and examine, my hypotheses in mice, are they also present in humans as well? Because ultimately, we want to help human patients with chronic pain. Some techniques that I use to study pain in the lab. So one of the main ones that I usually use is electrophysiology. So this is when I'm recording the electrical activity of these neurons. And the electrical activity of these neurons is very, very important. And it's a well-studied field in the entire field of neuroscience, not just in pain, because how these neurons fire, their electrical properties, are basically how they talk to other neurons. So it's almost like studying the language of humans is how you understand like the culture and everything else about civilization. Is really, language is a huge part of it. So the language of neurons is how they fire their electrical activity. And to do this, I have a setup. I have a microscope, so I can visually identify these really small neurons. And I have something here that is a glass electrode that is pictured schematically here. that I can seal onto these cell membrane of these neurons, so the outside of these neurons, and then I can use this electrode to sort of listen in on the conversation that this neuron is having with its neighbors. I can I can amplify this signal because these neurons are very small, so the electrical activity of these neurons are on the order of like millivolts, so they're very small electrical activity. I can attach this, this is attached to an amplifier that can then amplify this noise, and have it on my computer and examine these, spikes that these neurons, these action potentials that these neurons are firing. And looking at the different properties of these action potentials, I can see, is this neuron in a disease model, this activity will change, and it can give me information on how this neuron is communicating to its other neurons and how it's transmitting this pain information. So this is a technique that we use in the lab a lot, electrophysiology. We also use different stainings. So this here is a mouse spinal cord section, and shown in blue are all the different types of cells in the spinal cord. So you can see that this is just one section of the spinal cord. So this is already a lot. And shown in white are all the inhibitory neurons, and shown in green are all the excitatory neurons. So you can see that the circuitry of the spinal cord is very, very complicated. So how do we know, like in the context of chronic pain, like are all of these neurons changing? Are only the inhibitory neurons changing? Are only the excitatory neurons changing? Maybe only one population of the inhibitory neurons are changing. And if we're able to restore that change back to its normal, can we alleviate the pain? So we use techniques like staining to examine different expressions of certain proteins that are in neurons, to examine different expression of mRNA that are in neurons. And this way it can give us an idea of, what proteins are being upregulated or what proteins are being downregulated in the context of pain. And if the proteins that are being upregulated, if we can find out a way to sort of downregulate this protein back to the normal level, is that a possible mechanism to alleviate the pain? To study pain in mice, we have to use a model of neuropathic pain. So neuropathic pain is the main type of chronic pain that I study. And to do this, I do something called a CCI injury. So this is a chronic constriction of the sciatic nerve. So the sciatic nerve innervates the hind limbs of an organism. And if this is sutured, then it it constricts but not arrests the blood flow in this nerve. And this is to sort of mimic a nerve crush injury. So a way that people develop neuropathic pain is perhaps through a car accident or a accident where their nerves are severely damaged and it's crushed. So now they develop chronic pain in the form of different symptoms, such as hypersensitivity. So they could be more sensitive to the cold, more sensitive to touch, more sensitive to heat as well. So these are symptoms that patients with neuropathic pain have reported. So we use this model in animals to sort of try and replicate, or as close as possible, to what patients with neuropathic pain are dealing with. So I also have to test mechanical sensitivity in these rodents, because one of the main symptoms of patients with neuropathic pain is something called mechanical allodynia. So this is when light touch is perceived as really painful. So in a lot of patients with mechanical allodynia, just wearing a shirt with the tag on the back that's rubbing against perhaps their injured area is really painful for them. When they're outside and there is wind blowing on their skin, on their limbs, it becomes really painful for them, even though these sort of stimuli are not normally perceived as painful. So to measure this mechanical allodynia in rodents, we use something called von Frey filaments. So it's these filaments here that are increasing in force. So this filament here is increasing in strength. And what we do is we apply pressure on the mouse's paw, and we're able to measure the nociceptive or pain-like response of these animals at different filament strengths. And that can help us determine the threshold, the pain threshold of animals. And we have shown that in animals with neuropathic pain, this threshold is drastically decreased, because now a filament strength that is normally supposed to be like touch, that is normally supposed to be not painful, is now extremely painful in these animals models. So this is how we're able to measure and test for mechanical sensitivity in rodents. And these von Frey filaments are not only used in preclinical studies, they're also used in clinical settings as well, where people do these testings and try and measure mechanical sensitivity in human patients as well. So now moving on to spinal cord circuitry. So I have here a simplified version of the spinal cord circuitry. And here is the electrode that I showed previously, where I'm able to listen in and record the electrical activity of certain neurons. And you can see that just like a language, these neurons fire. Their firing patterns have many different types. So this orange neuron, for example, it has adaptive firing. So during a stimuli, it fires only at the beginning of the stimuli and when the stimuli ends, but it doesn't fire throughout the stimuli. However, you have, this blue neuron here that fires the entire time, and it shows something called tonic firing, where it's able to fire at very, very high frequencies the entire time and not sort of get exhausted. And then you also have neurons like the green neuron here that only just fires once. So what I'm interested in is, how does the firing pattern of these different neurons, different types of neurons, how does the brain perceive the pain? And also what happens in the context of chronic pain injury? So in the context of chronic pain, you can see that sometimes this blue neuron here is unable to fire at its very high frequency firing, and it now shows exhaustion, and it's unable to fire the entire time. So the specific population of neurons in the dorsal horn that I'm interested in is the parvalbumin or PV expressing neurons, shown here, and they are inhibitory neurons that receive input from the periphery. So these A beta fibers send touch information, and they're able to activate these PV neurons, which the PV neurons then in turn are able to inhibit their downstream neurons, such as excitatory neurons that we call them PKC gamma expressing neurons. So the circuitry, this is very simplified, because as you saw in the previous, where I was showing the staining of the neurons, there's like thousands of neurons that are just in one section of the dorsal horn. And what we're trying to do is give them specific, like to tease out the different subpopulations and see how these subpopulations react in normal condition and in chronic pain condition. And maybe that gives us a better idea of how the whole circuitry acts. So these parvalbumin neurons, they're expressed in the dorsal horn area here. So when I take sections of the spinal cord, they're closer to the back of the spinal cord. And you can see I have an image here showing the sort of labeling of these PV neurons. So it's these cells here that are being labeled, and they're really in sort of just below the superficial dorsal horn. So if this is the superficial side of the dorsal horn, they're just below, and they sort of make this sort of very nice line of a lamina of these neurons, and this is where they reside here. So we use different techniques such as transgenic animals, such as staining and labeling these neurons to be able to tease out just a very small portion of neurons compared to like all the thousands of neurons that are in the spinal cord. So in our previous work, in our 2015 paper, we showed that if I take a virus and this virus is able to selectively be expressed in these PV neurons, and they express something called an inhibitory dread. So this inhibitory dread is almost like a light switch, where if I switch it, if I activate this dread with something called CNO, I'm able to turn off these neurons, essentially. So you can see here that when I inject this virus, when this inhibitory dread is only expressed in the PV neurons of these animals, there is a significant decrease in the mechanical threshold of these neurons where their PV neurons are silenced. Of these animals where their PV neurons are silenced. So these animals develop mechanical allodynia where light touch is now perceived as painful for them. And you can see that this is shown by the decrease in their mechanical threshold. Now, what's even more interesting is if I then express an excitatory dread in these same PV neurons, and I do this in a neuropathic mice. So these animals already have very low mechanical threshold. They already demonstrate mechanical allodynia. I can slowly rescue and restore the normal mechanical sensation in these animals by just turning on these PV neurons. So you can see I'm able to alleviate the mechanical allodynia. So this is really interesting because it seems like just by boosting the activity of these PV neurons in these neuropathic mice, we're able to significantly help alleviate their mechanical allodynia in the nerve-injured animals. And one of the projects that I'm working on right now is looking at how do we boost the activity of these neurons in a more physiological setting. So these dreads are not normally expressed in these animals unless I inject them with a virus. However, these things called ion channels are normally expressed in all types of neurons. And depending on these ion channels, which ones are open, which ones are closed, which ones are more numerous in the membrane of these neurons, it would change the way these neurons fire. So by playing around with the ion channels and something called the ionic conductance of these neurons, I'm able to sort of play around with the firing activity of these neurons. I'm able to boost the activity of these neurons or diminish the activity of these neurons. And to do this, we can use things like drugs, like drugs that are able to block the ion channel, or drugs that are able to open the ion channel. And when we do this by modulating the firing pattern. So you can see that there's many different types of firing patterns for many different types of neurons. There's something that I mentioned previously. So these neurons that fire tonically very fast the entire way, these fast spiking neurons. There's also neurons that have a more irregular spiking pattern where they can fire, more in the beginning, and then they sort of space out a little bit, and then fire a bit more, and then a bit more spaced out their spikes, and neurons that have a more regular spiking pattern where each spike is evenly spread out, and then neurons that sort of almost seldomly spike. So they have one spike and then they don't spike for a very long time, and they spike again. So you can see that this is just 4 different types of firing patterns, but there are many different types of firing patterns, and this is how neurons communicate to their neighbors. So it's very important for us to understand how the neuron is able to communicate to its neighbors. And then if the firing pattern changes after nerve injury, then there's a miscommunication with the neurons. And that's why we get the symptoms like mechanical allodynia that are commonly seen in chronic pain patients. This is recordings from our lab that I've done in the past, where I was recording from PV neurons. And you can see here that in a normal condition, in an animal that does not have mechanical allodynia and is not in pain, these PV neurons are able to fire at very high frequency firing patterns and demonstrate the tonic, very fast spiking trademark here. However, after nerve injury, when these animals have a neuropathic pain and they have mechanical allodynia, you can see that these PV neurons are no longer able to fire at very high frequency firing patterns. So it's almost as if the language changed, and now they're communicating a totally different idea to its neighboring neurons. So now this miscommunication is what could possibly be one of the mechanisms leading to the symptoms of chronic pain because of this miscommunication of these neurons. So something that I'm really interested in is restoring this PV neuron firing. And I'm doing this by targeting different ion channels on these PV neurons and either blocking or activating them with different drugs to try and boost this activity of these PV neurons. Because, if after nerve injury, there is a decrease in this firing pattern and a miscommunication to its neighboring neurons, if I restore that communication and I restore that tonic firing to something of a similar level as a normal condition, am I able to help alleviate some of that pain from these animals? So this is a possible mechanism that we're studying in the lab that could be a potential therapeutic treatment to alleviating chronic pain. And so all of this work now is being done in mouse because it's a very easy animal model to work with. We're also very fortunate enough to get tissue from non-human primates such as marmosets, and this gives us one step closer in the evolutionary timeline to humans, and the circuitry in marmosets is much closer to humans than what we see in mouse. And also because we're fortunate enough to get donor tissue, donor spinal cord tissue, we're also bringing that back to the lab and trying to record from neurons in human spinal cord tissue to be able to see if the ideas and the research questions and the hypotheses that we have in mouse, if it's also relevant in humans as well. And this sort of closes the translational gap between species even more. So with that, I'd like to acknowledge the members of Dr. Reza Sharif-Naini's lab, so all of the people that helped, and our funding agencies, whom without none of this would have been possible. Thank you for listening, and I'll take any questions.